I'm so glad that I didn't scare you off with that first episode, which gave us the background information on our mystery pathogen. And today we're going to talk about the second thing, which is genome sequencing and quality analysis. Two admittedly dry subjects, but I will try to make it as easy and interesting as possible. During this webinar series, what I want to take you through is looking at the quality of reads, or how well the sequencer did or your isolation did, pathogen identity from those reads. We're going to then take those reads and assemble and annotate them to give us a genome. We're going to identify the pathogen from the assembly or from the contigs of that genome. And then we're going to look at phylogenetic trees. We're going to do a somewhat deep dive into phylogenetic trees. When one is doing genome sequencing, first you're isolating the nucleic acid, and then generally this is what you ship off to the sequencing center, where they do the library preparation, the cloning, amplification, the sequencing reaction, and then they generate these read files, which are called FastQ files. I'm not sure where that comes from, but computer scientists, they find interesting names for everything. Now, sometimes these FastQ files are of poor quality. It could have been your side. There could have been problems in the sample isolation. Maybe you got something in there that cut up the DNA and it's junk. There could have been problems with the sequencer. That sometimes happens. Poor quality reads, and I want to emphasize this, if the reads are bad, it can affect the downstream analysis, and it may lead you to make some conclusions that aren't necessarily true. So let's start out feeling confident that at least the sequencer went okay and you have good reads. So I know a lot of people are doing Illumina sequencing because it's pretty cheap and it's pretty fast and easy to do. And those reads come, as we mentioned earlier, as FastQ files. And so this, what you're seeing here in this lower part, is a read file. This is what it looks like. And in a FastQ file, there are millions of these things. So what does any of this mean? Well, this first line has the sequence identifier and the information about the run and what cluster it ran on. The second file is the sequence itself, and it's calling the nucleotides, which are A, T, C, and G. And sometimes it'll call an N, or depending upon the quality of your sample, it could be all N's. And that means that the sequencer couldn't figure out what it was, because at that particular location, it couldn't make a decision as to what it was. The third line there is just a separator that they put in. And then this fourth line are the base quality scores. So for every nucleotide in the actual sequence, there will be a corresponding base quality score. And that means something to the people who know a lot about these particular files. So these A's and E's and little figures are meaningful to them. So like I said, this is one read file. Imagine if you have millions of these to go through. How in the world could you possibly look at them, make a decision, and collate all that data? I don't think anybody can do it, but computers can do it. And so there are pipelines out there that you can use that are freely available that will tell you about the quality of your data. And that type of report is called a FastQC report. And it's providing a basic overview of not just single reads, but it takes all the reads in a FastQ file and summarizes them there. And so the one thing to remember, if nothing else, is that green is good and red is poor. So on your left, you have a good example where most of the reads are good quality. You can see that at the very end here, you have some tailing into the orange, and these are standard error bars. It's showing that some of the reads are down in poor quality, but some of them are also in good quality. So when you're looking, that's the one way that you can interpret this. And this, you can see that this guy really isn't very good at all. If it's in Illumina sequence, it's not a good one. So how can we generate a FastQ file? How does anybody do it? Do you need to do it by command line? Do you have to have a bioinformatician do it for you? No, there's a place that I like to do it, which is the Bacterial and Viral Bioinformatics Resource Center. It's a free resource that's funded by the National Institutes of Allergy and Infectious Disease. It's been funded continuously for 19 years, and they have a lot of bioinformatic pipelines at this resource that are all free to use. What I like most about it is I'm a biologist. I'm not a computer scientist. I can't do any coding. I can't do anything on command line. They have a very friendly user interface that helps the biologist upload their data and look at it. Let's do a quick demo. And this is the Bioinformatics Resource Center. It's at bv-brc.org. Now, the one thing they ask is that you register for an account. When you register for an account, you get a workspace with unlimited size at this moment, and you can run any of the pipelines. It doesn't cost you anything. And the main reason they ask you to do it is so that they can give you your own private workspace where you can keep your data private. Nobody else can see it. You would have to go here and do register, and they ask for your email. You have to have a username that you would use, your first and last name, and generate a password. But I'm already registered, so I'm just going to sign in. And then when I'm signed in, I've got the little avatar here, and you can see I use this resource a lot because I can't do any coding. I have to do everything here. And I've got almost 6,000 jobs here. So up here are the tools and services, and these are all the things that you can do at this resource. And we're going to step through a few of them while we're going over this series. But today, we're doing something to look at read quality. And to look at read quality, you need to do here under Utilities and click on FastQ Utilities. So we click on that. And this takes us to the landing page for that particular service. And you can see that there are parameters that they want you to do. There are pipelines. And if I click here, I can see there's a pipeline to trim. There's that FastQC. And there's also a pipeline that will align your reads to a reference genome if you're interested in that. But they accept paired reads, single reads, and those single reads could be Illumina, BacBio, or Nanopore. Now, why don't they have choices up here? Because all paired reads are Illumina reads. Or if one has data or is interested in data at the Sequence Read Archive, another free resource funded by the US government, you could put in the accession number here. But for this, I'm just going to step you through it pretty quickly. Let's start with the parameters. If I wanted to create a folder to do this, if I click on the down arrow, I can see folders that I've created recently. But if I want to go to my workspace, when I click, let me show you again, when I click on this folder, it's going to open up a dialog box with my workspace. And I could create a folder here by doing something like this, or I could upload data here, but I've already created this folder. So I want to go to the one I created called Pathogen Murder Mystery. And if I want to just save it there, I would click OK. So it's going to save it in that folder. I could have used one of those subfolders. It needs a name. And when I've done everything, when I can submit the job, this will turn blue. Mystery reads FastQC. Okay. So I've got a folder and I've got a name for this job. The pipeline I want is FastQC, which is doing the quality. So I click on that, and then I need to move it into this box. So I click on that plus icon and here it is. I could do a number of things here. I could trim it, I could map it to a reference, but all we're interested today is to see if these reads are any good. We don't want to waste a lot of time here. We want to move forward. So read files. I already have files here to analyze. So if I knew what they were, I could click on the down arrow and see ones I've uploaded recently, or I could navigate in my workspace by clicking on that folder icon. And then I have to navigate down to my folder. And there I've got the mystery read. So I click on the row and that highlights it. And then I can click the OK. And look, the name appears here. And I'd have to do the same thing again. Click on the folder icon. And I've got a lot of stuff in here. So I know I created it pretty recently. So I'm auto sorting by creating on this last column head. And then I click on this and but wait, before I do this, what if I wanted to upload something else? What if I didn't want to do this? You could click on the upload icon here and it's going to be looking in your computer. It's going to open a dialog box with your computer and look for read files. And those read files will have this ending at them. They're either in FASTA or FastQ format. So if I click on select file, it's opening that dialog box with my computer. And then look at this. It's only highlighting those files that are matching those endings, the FastQ or FastA. So I could say, this is the one I want to upload, and I could start that upload. But you know what? I don't really want to do this. So I'm going to cancel this. And this is the read file I want. So I'm going to click OK there. And I did it. Why isn't the submit button blue? It's not blue because I haven't moved my read files into this selected libraries box. You could run a bunch of these at the same time for different sets, pairs of read files. So it's helping you to add more in. But notice when I clicked here and moved this in, now the submit button is blue. So I can click on that and get the job started. And it says it's submitting it. And then it's been successfully submitted. Now, when I do this, or when anybody does it, you enter a queue with a lot of other people out there submitting jobs. And so it's on a fairness schedule and a first come, first serve, unless you're a big pig and you've entered a lot of jobs all at once. Often the command line people do this. We biologists, we don't do this kind of stuff, but the command line people will go in and swamp the queue by submitting a bunch of jobs at once. And so there's a fairness queue there to make sure that this doesn't happen. I can view my jobs by clicking on this jobs icon here. And you can see that it's off and running. Well, I can already see it because one that I ran earlier. So now I'm going to have more of them. But let's look at this one that recently completed. So when I click on that row, it highlights the green action bar with things that I can do with this particular job. And I want to view it. So I'm going to click on the view icon. And these are the job results. And it's also telling me what the job ID is, how long it took to run, 31 seconds. And these are the files for one of the reads and the other of the paired end that are both trying with the same piece of DNA, trying to sequence toward each other. So let's look at that. So here's read one. So I click on this and notice once again, when I click on it, the green action bar gets highlighted with possible downstream actions, and we want to view this. So I'm going to click on the view, and here it gives me some information. It says that these were Illumina sequences. It tells me how many sequences there were in this file. It was more than 3 million, almost 4 million. Are any of them poor quality? No. What's the average length across all of them? It says 101 base pairs. And the GC percentage is 50%. So look at this. This actually looks pretty good. You've got some dipping down. It looks like there's some problems with the sequence a little bit. Still, I think this looks great. I'd be thrilled with this. I'd be thrilled to continue with this. Notice that this is talking about the 1 to 100. And so it's looking at the first, second, third. It's taking all those almost 4 million reads, and it's summarizing the information for all of them at each of these sites. And you have a standard error bar to show you where things are going with that. And then you have per sequence quality scores. Let me point out at the side, if it's got a green check, it means this looks good. If it's got this exclamation point in yellow, it means, well, there's not a lot of concern, but a little bit of concern. And when it's bad, it'll have a big red X, and we don't want that. So per quality sequence scores, that's looking across each of them, and that's what you want to see, or that's what they say you want to see. The sequence content, sometimes this has a lot of noise going in, but this is nice and tight. And this is the one where they have it. The theoretical distribution is blue, so they have a warning sign. The theoretical distribution is blue, and the red ekes out of that just barely here. So that's a little bit concerning, but I'm not going to be concerned about it. I don't even need to look any farther because I'm going with this data, but you're new to this, so we'll go all the way, and I'm doing it just for you. It's looking to see if there are any ends in the sequences next, and it's not finding any. It's looking for the length distribution, and this looks good too. And it's looking to see if there's any duplication in there, and that looks good. And this one, did we see any overrepresented sequences? No. Did we see any adapters that would need to be cut off? No, we don't. So this is all good. This is a breadcrumb at the back, so I can go back to my job folder, click there, and let's take a quick look at mystery read 2. And we need to click the view icon, and then, well, this one looks good too. This isn't bad. Look at that. Sometimes you'll find when you have paired in reads, one will be good and the other will be kind of minimal quality. Both these look good. We should be thrilled. So as far as I'm concerned, we're going to be good to go with this. Let's go check out where we have the warning sign. Looks pretty similar to the other guy. Okay, so I think I'm going to give this 2 thumbs up, and I think we can proceed to the other steps. But let's go back to our slides because I can't let you go without boring you just a little bit further. We did the demo. I showed you how to do it in BVBRC. And the mystery pathogen read quality, good. We like it. There was a little bit of a warning with this one, but nothing I'm going to worry about at all. And so I invite you, because now we're starting to get into the good stuff. And I'm going to show you how just from those read files, you can submit those to bioinformatic pipelines. You can do it. You don't need a bioinformatician to do it or a computer scientist. You yourself will be able to submit those reads to a pipeline, and it will give you a guess of who they think is in there. So I appreciate you joining me, and I hope that you will come along for the ride for the next one. Thanks a lot.